Welcome back to our 5th episode of OMg OMx. I'm starting to notice a trend here, and that's that some of the successful people we've been talking with are all collaborative, inquisitive, and trying to do science that works for them. Hi there, Balyn. I'm so excited that you've agreed to be our 5th podcast guest on the OMg OMx series, and I'm excited for you to share your story with our audience. So maybe you can start by telling us a little bit about you and how you got to your position and why we're here today.
Sure. So I started my scientific journey as an undergraduate researcher in an organic chemistry lab. And during this time, I was really actually passionate about like engineering and chemical engineering, and I wanted to work on this like particular methanol fuel cell project that was running through this lab. And so when the faculty member approached me about signing up for a PhD in chemistry, I didn't really think too much about it because I was thinking more about working on this fuel cell project. And it seemed like a pretty awesome deal to be able to like get a PhD in science. At the same time, I was working on this like research project that I was super excited about. And so I signed up to be a PhD student and basically joined during the financial crisis of like 2008, 2009. And promptly the funding for the project was cut. So I found myself in a PhD program in chemistry with this intention of working on like fuel cells. And that really prompted me to start thinking about what I would do next, as I was amidst a financial crisis and now not knowing what was around. I decided to give myself a year to figure that out. And I ended up connecting with a brand new assistant professor who was starting their lab, who's a chemical biologist. And I thought it was pretty cool to be able to make molecules that then could ask biology questions. And so I ended up starting in his lab and getting super excited about kind of chemical biology. And we started making chemical tools and wanting to do mass spec with them. And that was really awesome, but I was always sending samples out. And so when I was a postdoc, I decided I wanted to go to a lab that would allow me to understand what happened to my samples after I sent them away. And that's when I went to a chemical proteomics lab and worked there for a few years, but also wanted to gain additional kind of research experience in a specific field of biology that I felt was understudied using the tools that I was super familiar with. So I moved to do a second postdoc at Stanford. And I was there for 2 years I was there for a little over a year, actually, when a school reached out to me and asked me to apply for their faculty search. And at first I thought, oh, no, I'm not ready. But my PhD advisor reached out to me and said, when a school does that, like, you apply. And UCSF was the only other school that still had a job opening because it was so late in the cycle. And so I applied to both schools, wrote my job application in 1 week, which is like not what anyone would tell you to do. And I ended up having the privilege of being able to come here to UCSF and start my lab, where we're really merging chemical biology and mass spec to answer questions in the field of innate immunity.
So it sounds like with so many scientists, you fell into a number of different things that helped you to figure out your path and to move along that path to get where you are now. Who are some of the people that have been influential to you and who've helped you along this journey so far?
Sure. I think probably the most influential person has been my PhD advisor, his name is Matt Pratt. He's a professor at University of Southern California. He really kind of like took a huge risk in taking someone who only had organic chemistry experience and really set me on this path of wanting to answer important questions in biology using chemical tools. And I think that I kind of entered everything with the idea that I had nothing to lose and I would just say yes to things that I was curious about and that interested me, and to not think too much about it, but just to say yes to all of the opportunities that seemed worth exploring.
That's great. And I think without good mentors, it's hard for any of us to progress. And to see you now as a professor passing that along is critical and important for our educational system. How do you feel about being in the San Francisco area where you have so many opportunities? You're in this hub. How does that influence your ability to be a good mentor and to be a good scientist?
I love working in the Bay Area. That was one of the reasons that I wanted to come to UCSF, because I feel like when I got to Stanford, there's just this energy about the Bay Area where it really feels like anything is possible. You know, like the person in front of you at a coffee shop might be the founder of like the next big tech company or an amazing biotech that has some new revolutionary treatment for people or diagnostic. And I think that it's an awesome place to learn to be a scientist as well for trainees, because there's such an ecosystem here and such a strong relationship between industry and academia and venture capital, that really, no matter what you want to do next, there's kind of an opportunity here. And I think that kind of healthy energy of being able to not feel stuck in one place or to feel like you're in like a college town or in a place that has an identity other than like exactly what you're doing in that moment is like a pretty awesome place to be.
You're making me super excited. I feel like I need to come visit now and see everything that's happening and who might be in a given coffee shop at any moment.
It's also a really beautiful place. Like, sometimes I just like drive up a giant hill in San Francisco and you can see like the Bay Bridge and the Golden Gate Bridge. And it's just like unbelievable that there's this major city on top of a million hills, like with water all around. So it's definitely a really beautiful place to work too.
So you seem to draw some inspiration from your physical surroundings. So what about if we take another step back into your training? What was one of your OMG moments for mass spectrometry? You said that you wanted to go to that type of group to see how all this was done. When did you feel like something was changing and turning in your mind?
Sure. So I think that my OMG moment was probably when I made my first chemical probe and we treated cells with it, and then we looked to see what proteins were modified on a fluorescence gel, and that was really amazing to see something that I had physically made like incorporated into cells and be able to give us some sort of information. But the information was like very qualitative of like there's this much more signal over background, or maybe there's this much more of a change in signal over what a healthy cell looks like versus a disease cell looks like, for example. And so when I realized that we could actually prepare these samples in a way that would tell us the identity of who they were and how things were changing, that was super exciting. So being able to like send my samples out to a mass spec facility and get back this like slew of information of like what proteins we were actually modifying was super exciting.
So I think, again, you bring a different perspective than some of our former guests. And you're a chemical biologist by training. You look at things a little bit differently than a traditional chemist or mass spectrometrist. So can you comment maybe on how biologists have found interest and utility in mass spectrometry and how this tool is still so important to be spread broadly?
I think for so long, this is probably like a hot take for mass spectrometry, but I think for so long, it was such a feat to be able to identify proteins or modifications or what have you in your mass spec sample, that it felt like there was this disconnect between the technologists who were developing these instruments and the biologists who maybe would at some point want to use them. I feel like as the technology has improved, the biologists have still been fearful, right? Where they have this idea that they need, 50 million cells and they need to have all of these very, very specific tools to be able to ask questions that they're interested in answering. I think that we're now at this point where the technology is at such a place that biologists don't need to be worried, and that they really can leverage this technology to ask important questions. And so my lab is really interested in kind of helping to bridge that gap to sort of say, like, we are bona fide biologists who have a very specific question we're asking, and we want to develop technologies that other people who aren't mass spec experts can use, so that they can leverage us as well.
So, what else has been really important in that journey, in that process? You say the mass spectrometer itself is easy to overcome, learn, and do what you need to do. So, where does the challenge lie for you, and what tools have you used to overcome those?
Sure. So I think there's been two major challenges. One is with kind of the software and data processing and analytics. And the other has been with sample prep. And I think with software, we used to used to have to inject so much sample, and then you would get these ratios of like what percentage of things that you inject on the instrument can actually be attributed to a protein or peptide. That ratio used to be very low, right? And not like a long time ago, like 10 years ago, 15 years ago. And so I think there was also this idea that we were losing so much information. And I think the instrumentation has improved in terms of efficiencies and in detecting things and being able to run MS1s and MS2s and sometimes MS3s. But then the software leaves a lot to be desired sometimes. And the software really has improved recently. So now we're getting percentages like between 30 and 50% of what the mass spec is detecting can be attributed to a protein. And so I think that's like really exciting. And there's obviously more that can be done to get even higher. But I think that those are massive strides to have made it in a relatively short amount of time. And then in terms of sample prep, there are these like very beautiful technologies that have been developed, but they're pretty inaccessible. And so I think that companies that are making kits to be able to essentially process small amounts of sample have really sort of paved the way for this to become a more accessible technology for everyone to use.
So, I'm pretty sure that you use PepSap and that you use Proteomics for some of your sample prep. What do you hope to see from those in the next 10 to 15 years to make your job even easier for doing, the front end work and then letting the mass spectrometer do its thing?
So with Proteomics, Proteomics kits are amazing. They essentially let you mini prep mass spec samples. So if you can mini prep DNA, you can prep a mass spec sample and have like a very high quality, clean sample. And they've done a really great job of sort of creating a accessible technique. I would love to see them go lower so that maybe everything to miniaturize their sample prep so that we can prep even less. Right now we can inject like 150 nanograms of a complex proteome and get back 5,000 to 6,000 protein IDs. But we have to prep about 1 microgram of protein in order to recover enough protein to do a few replicates. And so I would love to see things go even lower. And I think it would probably just take like miniaturizing some of the cartridges that they use. PubSub columns have been a huge game changer for us on the kind of the chromatography side of things. I think because they are a little bit wider, we run them at lower pressure, but the separation is phenomenal. They're reasonably priced and they last a long time. So we find ourselves replacing a lot more of the kind of fluidics connectors and tubing and the emitters much more often than we actually do our columns. And so I think that also has been a big game changer for us because when we have really precious samples, like when you throw a clog in these kind of like these smaller, higher pressure columns, it's kind of game over. And the quality and reproducibility of the column is also great. So before, when we were using other columns, we would have to try like 5 or 6 of them to get one that just had really beautiful chromatography. And when it worked out great, that was awesome. But then you throw a clog in, you have to start all over again. And so the PubSub columns are just real workhorses. They've really, I think we've gone through 3 in a year and a half, which seems totally reasonable. We're running like a few thousand samples a year.
So what about the learning curve there? Has it been easy for you to onboard these? Have you needed a lot of support from technicians, from application scientists, or have you just been like off and running?
We were able to get methods from application specialists through Bruker, and we really haven't changed much beyond that. So it has really been a plug and play setup for us for the most part.
Okay. So why don't we kind of go back to some of your science and some of your experiences that you've had? So as a chemical biologist, you're clearly interested in proteins and proteomics, but what is the critical information that you're getting for your innate immunity research?
Sure. So we are really interested in kind of two main questions, which is what makes macrophages, which are the kind of central innate immune cell in our body, decide to eat another cell or not eat. And so that is really a question that we are getting at. We are trying to develop small molecule modulators of these processes. We're trying to understand how these processes are altered in different disease states in different individuals. I think when we think about it from a therapeutic perspective, in trying to develop new therapeutics in this space, but also trying to understand why only a subset of individuals respond to therapeutically targeting this process right now.
So what do you think that the big necessary outcomes in the field of innate immunity research are in the next decade or so? What else do you need in that regard to be able to make advances and make an impact on your research?
For us, I think there's a few things the innate immunologists may disagree with me, but I think that this space is just broadly understudied. It wasn't until a few years ago that the NIH required that people use equal numbers of male and female mice in experiments, for example. And oftentimes, and we know that there are there are sex-based differences in the innate immune system, and this has been widely reported, but it hasn't necessarily been exhaustively characterized. And I think that there's been these massive efforts for like RNA sequencing to look at different cell types and their signatures. But we still don't have a really good understanding of the proteins that are expressed by all of these cell types. And so I think as technologies like mass spectrometry become more widely adopted and we've really hit this point now where I think we can get so much information out of the experiments that we do that we'll have a better understanding of what these cells are actually capable of doing, and then hopefully move to also studying how post-translational modifications, for example, also regulate them. But I think there's a wealth of space for like a ton more science to be done. I think like I had a student who I met with last week, and they were saying, I'm just unclear what direction I should take my research in. And the reason for that was not because they were stuck at a spot that was, overstudied and they didn't know how to differentiate themselves. The problem was really like, there are so many directions to take. There's so much that's understudied. So how do I choose?
That's an exciting problem to have. That's much better than the alternative, I think.
Oh, totally. Yeah. I was like, this is a great space to be in.
Absolutely.
But think about experiments that'll help. Like you have the privilege of being able to really, you get to pick what are you driven by. And I think that to be able to be in a space in science where you're not confined to what is already known and how do I create a niche for myself is pretty awesome.
It's a huge luxury. Yeah. Like you said, it's a huge privilege. So let's go back to numbers for a minute, because when we chatted previously, you were just kind of blowing my mind with, how many cells are in various systems. So I took a couple notes and I wanted you to remind me and see, if we can just throw out some fun facts here. So when you have a really small and precious sample, what exactly are we talking about here? So you have a microgram of cells, but depending on cell type, that's going to vary the number you have, right?
Yes, absolutely. So if we think about like hematopoietic stem cell, which is a blood-forming stem cell. These are super tiny cells. They don't have a ton of protein because they don't want to commit to a specific fate. And you need about 35,000 of these cells to get 1 microgram of protein. If you think about a terminally differentiated cell, like an immune effector cell, like a macrophage, macrophages are super protein rich, and we only need about 3,000 cells to get a microgram of protein.
So again, in your method development, in your experimental planning, you've got to really think more about the biology here to be successful and not just your prep and everything else.
Yes, absolutely. And especially, when you think about purifying cells and you have 3,000, for example, your sample has to be so super, super pure, because having, 80% purity wouldn't be enough in this case, right?
So let's maybe tie this back to where you had some of the most fun in your experimental planning, in your actual experimental endeavors. So you've done a lot of collaborations, you've done a lot of one-off studies to help people because you've really become this queen of sample prep that can do what's needed. So what are some of the more fun experiments or samples that you've worked with that you could share with us?
A pretty fun project that we recently completed and was just accepted at Nature is that we have a neighbor on our floor at UCSF who studies calcium channels. And he came to us and said, we have this protein complex or protein degradation. We don't really understand what's happening here, but we are interested. We're doing a bunch of cryo-EM. And what we've noticed on our grids is that we have the structure of the channel, but we also have this like mystery density, and we don't know what it is. And he said, it could be another protein. We're not really sure. And he showed me a gel of what it looked like. And it looked like there were all of these bands. And the protein's quite large. So I thought, oh, maybe this is like degradation. I don't know. But then he told me that this protein had gone through 2 orthogonal purifications. And so I thought, whatever this is like pretty tightly bound to one another. And so we decided to do mass spec. And he just gave us the sample, we processed it, and my student Jose searched the data, and we looked at it, and we were like, this looks like, this is definitely, like, without a doubt, the EMC, which is this complex that has been like the holy grail in like membrane protein biogenesis. And the structure of the EMC had been solved before, but not with a client bound. And so what we ended up identifying is that this lab, or Dan Miner's lab here, had solved the structure of the EMC bound to a client, which gave us a ton of information about how these channels are formed and how they move to the membrane. But we also had all this beautiful quantitative mass spec data to show how different parts of the channel are sort of assembled together, and what the critical interfaces are. And as we lose members of the EMC or different parts of the channel, how that interaction alters. And so we could really quantify the interaction really rigorously using mass spec and cryo-EM together, which was like super fun to be able to like bring these technologies that have been changing, biology, but really bringing them together and like leveraging state-of-the-art techniques in both spaces. So that was super, super fun.
That sounds really fun. And I can see, you're asking really interesting questions. You're in this wide open space and you're going about things so cleverly. And I can see that in your accolades. You recently won the Beckman Young Investigator Award. Can you maybe tell us and maybe give some inspiration to some of the trainees out there on how to stick to this and how to make this work and build yourself a career that you're enjoying and proud of?
Sure. I don't really see myself as as like someone to look up to, but I think I have always, being able to do science is a huge privilege. We get to study things that no one understands or knows about, but hopefully you feel like is an important question that can, improve people's lives. And I have just always, I mean, in the same way that I stayed in grad school to see if there was something that interests me. Like I've always just kept going until I'm not having fun anymore. And so I think science should be fun. It should be exciting. And I just I'll stop being a scientist when I'm not having fun anymore. And I think that's like what keeps me going. It's amazing to be part of such an awesome community of people here. I feel like coming to UCSF, one of the reasons I wanted to come here was because I felt like it would elevate my research program, but also that I had something to bring to the community. And so being able to have collaborations like with Dan here has been awesome. He is also a former Beckman Young investigator. So that is pretty fun to be able to have this very fancy and humbling award, and to be able to share this with someone that now we're collaborating with is pretty cool.
Yeah, Balyn, you're doing fantastic. Own it.
I collaborate with people that I have fun talking to who are working on a problem I think is interesting and a problem I think that we can be helpful in solving. We have our own research direction and our own research identity, the questions we're asking, but I also I don't want to be so pigeonholed by our research identity that I turn down opportunities to expand what that definition is.
It's fantastic. So is there anything else that you'd like to leave as a closing message? If there's anything you'd like to leave as a closing message, this would be your opportunity.
Sure. I mean, I guess the one thing I would say is that there's been a lot of negativity surrounding career academics. And I really have to say, to me, I feel like I have the best job in the world. I get to work on things that I think are super interesting. I get to bring people into my lab who share common interests and maybe bring different perspectives to the questions that we're interested in answering. And I get to work with other like-minded people. I feel like I work hard, but I also don't feel like my life has had to suffer because I've committed my entire being to being a professor. And I think that that's the narrative that people see is in order to have this job, you have to sacrifice everything. I think there's a tremendous amount of kind of flexibility in your schedule, and I've had a child since I've been here, and I just think it is a much better job than say Twitter has made it out to be. And so what I would say to trainees is you shouldn't be afraid of this job, and it shouldn't be something that you think is inaccessible to you unless you want it bad enough to lose everything else.
So at the risk of perhaps tanking our own viewership here, don't believe everything you read on the internet, but maybe if there's an expert talking, we can tune in and be okay with that.
Or just do whatever you want and stop listening to reading people's tweets. It's a used car thing, right? If you have a bad experience buying a used car, you'll tell more people than if you had a good one.
Yeah.
So I think that is kind of the fate that academia has suffered. But
yeah, let's apply some psychology to our internet usage. Exactly. I hope you enjoyed our episode today with Balyn. I've really started to notice with proteomics that this is such a broad and huge field that needs researchers doing many different aspects. And today's episode brought in the importance of sample preparation to be successful. I hope you enjoyed, and I'll see you next time.